Okay, this is chapter 2 of Structure and Function, Introduction to Anatomy and Physiology, Module 1 on cells, a little biochemistry, organs and tissues. Oop. Okay, here are your learning objectives, come from the syllabus. So these are our goals. Okay, organization of the human body. So essentially we start out as on the very basic level. We usually start out at the cellular level. So a cell, and we say one cell that functions together, or many cells that function together become a tissue. Many tissues that function together become an organ. Many organs that function together become an organ system, and many organ systems that function together become an organism. But backwards from a cell is comprised of organelles, macromolecules, molecules, and finally the atom is the most basic unit for life. So here is another way of looking at this, with cells being the beginning, cells to tissues, to organs, to organ systems, to organisms, and the organism being the human body. Okay, I'm going to go ahead and play this video, kind of putting it all together. Oop. Not sure what happened there, but we could try that again.
I like to think of life science as one big pyramid. Let's call the pyramid biological organization. Let's work our way from very small and specific concepts to very large and complex topics. So let's start at the tip of the pyramid with something so small it can't be seen by the human eye: a single cell. A cell by itself is the most basic component of life. A single cell, which is capable of living on its own, is called a unicellular organism. Organisms like bacteria or protists. These organisms are everywhere you look. You just can't see them. But when cells join together with more than one cell like itself to perform a similar function, it's no longer a unicellular organism. It's then called a tissue. No, not that kind of tissue. There are 4 types of tissues found in the human body: connective, muscle, nervous, and epithelial. Bone tissue is a type of connective tissue, and it's not flimsy like a Kleenex at all. Blood is also an example of the same cells working together to perform the same job, but it's a liquid, again, not like a Kleenex at all. No matter what the consistency is, a tissue is a group of the same cells working together to perform a similar function. Anytime there are different tissues working together, we then call it an organ. That's the next level of the pyramid. Animals aren't the only ones with organs. Plants have organs too. When we find multiple different organs working together to perform the same job, we then call it an organ system. Take the digestive system, for example. It's made up of a mouth, esophagus, stomach, pancreas, liver, gallbladder, small intestine, and large intestine, also known as your colon. Although each individual organ in this organ system does a very different job, together, these organs work to achieve the one goal of taking all the nutrients out of the food we eat and getting rid of what we don't need. An organ system, by definition, can only do one job, like get the nutrients from our food, send and receive electrical signals, or exchange oxygen for carbon dioxide. Regardless of their individual function, all organ systems rely on one another to do the vital jobs which they can't do themselves. If one organ system doesn't work, the others will shut down too. An organism is a complicated living thing which requires the proper functioning of multiple organ systems to maintain stability. This stability is called homeostasis. When all organ systems are working together and maintaining homeostasis, then it results in a happy living organism. Organisms are incredibly diverse and can be as simple as a bacteria or as complex as you. When a group of the same organisms, also known as a species, live together in the same environment, we then call it a population. Often, there are several populations all living in one environment. That is what we call a community. Each time you look out the window, you're seeing a community. When we look at multiple communities, along with how all of the organisms interact with the physical environment, then we refer to it as an ecosystem. When you look at all the ecosystems of Earth together,
that goes a little further than we need to go. At the organism level, it would be fine for you to know. Okay, so you've got elements, atoms, molecules, and compounds. Let's dissect this a little bit. If you've not had chemistry, we're going to give you a crash course in basic chemistry. If you have, we're going to dust off your brain and try to remember what this is. Okay, this will help us. An atom, start with that one, is the smallest unit of an element. And think of an element as like a type, a brand name. So it is the smallest unit of matter, or atoms, that retains its unique chemical properties. So if the atom is the basic unit for life, or the basic unit, an element would be like a hydrogen atom, an oxygen atom. It's the type of atom it is. Two or more of the same atoms that are joined together are called molecules, and two or more different elements, chemically bonded and a fixed ratio, are called compounds. So water, really, because it's H2O, is a compound. 2 hydrogens, 1 oxygen. An atom, again, is the smallest recognizable unit of an element. And within an atom, you've got some protons, which are the positively charged particles. Neutrons are neutral. And electrons are very, very tiny, and they are surrounding the nucleus that contains the proton and neutron. They're flying around that nucleus, and they are negatively charged. Elements, you remember this, if you could dust off your brain a little from high school chemistry, this is the periodic table of elements. You do not need to memorize this, but whether matter is living or non-living, all the elements found on Earth are located on this periodic table of elements. So the smallest units that contain copper, for example, Cu, would be copper element, the copper elements. They cannot be broken down into smaller pieces. For your purposes for this course, it is very important that you understand some of the important trace elements in the body and what they're for and how we find them in food. So iron is Fe. That's the periodic symbol for iron here in the center here. Iron forms hemoglobin, which is necessary for oxygen delivery. And iron can be found in green leafy vegetables, in red meat, plums, cherries, egg yolks. Copper, Cu, helps with enzyme synthesis systems and formation of red blood cells and collagen. And that's found in almonds, peas, dried beans, prunes. You can see the list. Zinc, if you take zinc before you're getting a cold, it helps with immune system health. Manganese, again, the enzyme systems, your central nervous system, metabolism, blood clotting. Iodine is one that's very important for thyroid activity. We eat salt that is iodized, so we don't usually have an issue with this in the United States, but iodine is found in fish, in kelp, in seaweeds, and in onions. Fluorine is necessary for dental health, and fluorine is found in tap water. Here's some other ones that are kind of biologically significant. So you've got bicarbonate, that is a buffer that helps us with acid-base balance. You've got calcium for bones, carbon dioxide, chlorine. Here's just a few of the kind of important ones. And this is found from your, this is actually not found in your textbook, but important for you to know. It's one of the standards. Okay. Ions. Ions are when an atom is, normally an atom is neutral. Normally, the number of positive protons equals the number of negative electrons. But if an atom gains or loses an electron, it carries a charge. It can be positive or negative. And positive and negative act. And negative repels negative, positive repels positive. So if they're opposites like this, positive being a cation, negative being an anion, they're going to attract together. Okay, if there were two positives, they would repel. Okay, a little bit about bonding. And most of the reason that you need to know a little bit about bonding is because of the properties of water in the body. So covalent bonds happen if electrons are shared in a bond. So the key word here being shared. And a lot of times in covalent bondings, we have double bonds. In the case of a polar, so what does polar versus nonpolar mean? Polar means that there's an unequal sharing of the electrons. And nonpolar means it's equal sharing. So there's no difference in the charge around the molecule or the compound. Okay, so in polar, you've got more positive down here than you have negative up here. It's an unequal sharing of charge. And water is polar. Ionic bonds happen when one atom donates electrons to the other atom. So it's giving it. And because one loses it, the other gains it, the atoms carry either a positive or negative charge. So in this case, sodium. Sodium gains, loses an electron, it becomes Na plus because it's positive. Okay. These molecules are polar because the charges are unequal. Okay, all of that to say, the important reason you need to know that is because of water. Water being the chief liquid in all biological systems, every body fluid is water-based. 60% of an adult is water. 75% of an infant's body is water. Water is polar, it's covalent, it's hydrogen and oxygen with an unequal sharing. That's your take-home message. Water is necessary because we need it to act as a solvent for reactions to happen, transportation of material, absorbing, releasing heat, and functioning in the body as a lubricant. We won't get too much into this right now, but the body and the regulation of water and the amount of water in our body is one of those homeostatic processes. Okay, hydrophilic. So hydro meaning water, philos means love. So it's water-loving molecules. So if something is hydrophilic, it carries a charge and is polar and will mix with water. So in other words, salts, sugar, we can make sugar water. We can dissolve sugar or salt in water and it's hydrophilic, it will become a solution. Okay. Hydrophobic means like the top of a car, wax, means it's scared of water, water fearing. It does not carry a charge and it does not mix with water. So an example of that would be like salad dressing with fats and oils separating. Okay, so we kind of alluded to this, but solutions. We have solute, which would be like your salt or your sugar. You have a solvent is the liquid that you're dissolving it in. So in this case, water. And then the solution is the end product. It's the solute dissolved in the solvent becomes the solution. And the concentration of the solution is how strong it is, how much solute is dissolved in a solution. Okay. Tonicity. So tonicity of a solution can affect the volume of a cell. And one of the reasons that this is important for you to know is what happens to a cell with tonicity, also IV fluids, and different processes of the body to maintain its water balance. So this would be isotonic, which means its solvent to solution ratio is equal, iso meaning same. Hypotonic means it's a weaker solution, so it's not as much solute. Hypertonic means it's more concentrated. Okay, so this is what happens to blood cells. If we put a blood cell in a hypertonic solution, so if you remember that hypertonic solution has a lot of concentration in it, it's strong, okay? What happens is the water with osmosis wants to go from the area of low concentration to higher. It wants to kind of dilute that strong solution. So if we put a red blood cell in a hypertonic solution, water gets pulled from the cell and shrinks the cell. And that's when you end up with this like prune-like red blood cells. Okay. If we put red blood cells in a hypotonic, so the weaker solution, then the concentration is stronger inside the cell. So water goes from low to high and bloats those cells. It makes them fat. Okay. And isotonic would be the same. Okay, pH. pH is the amount of acidity or alkalinity in something. Okay, has to do with hydrogen ions and hydroxyl or OH negative. Okay, for our purposes, let's think acid base. Okay, alkaline is base, acid is acid. Okay, so neutral is 7 on a scale from 0 to 14 or 1 to 14. 7 is neutral. Our blood is 7.4 or a tight range between 7.35 and 7.45. So it is slightly basic or slightly alkaline. The higher the number, the more basic. So you've got eggs and bleach and hand soap being over here in the very, very alkaline or basic. Okay. The more acid things, think like lemons and tomatoes and vinegar. The more acidic, the lower the number. So the stomach acid is a pH of 2. If you vomit outside of a car window, the acid in your stomach will peel the paint off if it's left on there. Not to be gross, but that's a good example. Okay, here's another one. So basically, acids are chemicals that release hydrogen ions. We saw that, hydrogen ions, okay? It can dissolve metals. Acid can dissolve metals. So think about that the next time you're drinking a soda at a pH of 3. That can hurt your stomach. It's a little bit on the acid side, okay? We would never eat a battery, right? They taste sour and they have a lower number for pH. There's our blood at 7.35, 7.45, okay? Bases accept hydrogen ions. They taste bitter and they have a pH of greater than 7. So there's your antacids. There's a basic, baking soda is basic. Okay. An acid-base balance in the body, real quickly, the respiratory and renal or kidneys systems help us regulate. Remember, we go back to homeostasis. If we are too acidic, we will blow off carbon dioxide, which is acidic. We will hyperventilate or exhale too much to blow off some of that carbon dioxide, okay? The reason for breathing into a bag is to inhale some of the back of the carbon dioxide. You're blowing off too much acid, okay? So kidneys also do the same thing. They remove excess acid in the urine and secrete bicarbonate, which is a buffer, which the buffer brings things back to homeostasis. Okay. So I mentioned this, the body, it's normal pH. It's very funny here, 7.35 to 7.45. That's our normal. If we get alkalotic, we die. If we get acidotic, we die. So anything, it's a very, very tight range. Okay. Organic compounds. Organic, think carbon. Things that contain carbon are organic. Okay. And mainly there are 4 categories. You have your carbohydrates, your lipids, which are fats, proteins, and nucleic acids. And I'll go through each one of those. So inorganic, not organic, no carbon. So water, inorganic. Oxygen, inorganic. Oh, carbon dioxide. I might call that organic. Okay. We'll keep going. Inorganic would be, here we go with that carbon dioxide. Here it is. It's inorganic, usually don't contain carbon, okay? And it says here, organic is usually bonded. The carbon in an organic molecule or compound is bonded to hydrogen. So that's where the carbon dioxide kind of slips in there. That does have carbon. So salt, quartz, carbon dioxide, hydrochloric acid, excuse me, those are all inorganic, where these things are organic. Okay, carbohydrates literally mean watered carbon, hydro and carbon. And their basic formula is CH2O. It's a water with a carbon in some kind of ratio. And typically these are sugars and starches. These are energy sources. Monosaccharides are simple sugars, meaning mono, they have one sugar molecule, mono 1. Di have 2, and an example of that is your sucrose, your maltose, your lactose. 2 linked monosaccharides. Poly, you can imagine, mean many, and that's more of your starch, your glycogen, your cellulose. And glycogen is important for life because it's stored in the liver and we can release it to increase blood sugar if needed. Your lipids, okay, this is kind of a cool picture. So you've got hydrophobic heads and hydrophilic tails. And what happens is the hydrophilic tails kind of face each other and it makes like a barrier. And lipids or fats are made of carbon and hydrogen. And they're hydrophobic because they contain very little oxygen. And lipids or fats are great for energy storage, communication, and protection. Okay. So we have fats and oils. We have waxes. Think of a car, right? Hydrophobic. You have the water beads on top because it's hydrophobic. Phospholipids, there's the word in there, and steroids. Your proteins are made of long chains of amino acids. And you'll know a protein when you look at it because it has nitrogen as the backbone. And proteins we need for structure. proteins build things. They are found in our collagen, in our tendons, in our ligaments. They also speed up biological reactions in enzymes. They can store energy. They move the body in the form of muscle protein. The antibodies are proteins, and they protect against infection. And lastly, they allow the cells to communicate. Insulin is a protein with a nitrogen backbone. Your nucleic acids are essentially RNA or DNA. They're very simple, RNA or DNA, which contain genetic code. They control the activities of cells. We're not going to get too much into RNA and DNA. Okay, metabolism essentially is all of the chemical operations going on within our bodies. It uses energy and produces waste product. So reactions in our body can happen faster if the concentration of those reactants are increased. If we add heat to speed up the reactants, or a catalyst like an enzyme is used to speed up reactions. So that the metabolism is essentially all the chemical operations of the body. Cellular respiration, I'm going to go over this kind of briefly and hopefully superficially because this is a little complicated. The body uses energy in the form of ATP, which is adenosine triphosphate. If you know ATP, that's fine. One of the most important chemical reactions in our body, or cellular respiration to make energy, is C6H12O6, which is glucose. We take glucose from food, add it to oxygen that we're breathing, and what we expel is carbon dioxide, water, and a whole lot of ATP. If you can see it in the picture here, 36 ATP. And we need ATP to live. So we need glucose to live. So the reactants of this equation, the ingredients per se, are the glucose, C6H12O6, and the oxygen, O2. The product is ATP energy, that's the goal, right? Carbon dioxide and water. And that's a simple understanding of cellular respiration, but this is what we need our cells to do to produce energy and work. Ooh, my video is no good. I'll be sending that out to you. Okay, the last part of this is cells. Cells are the basic, fundamental, microscopic unit of living things. And cells have a lot of components. They have different sizes, different shapes, different functions, whether or not they be nerve cells, blood cells, whatever. Your cytoplasm is gel-like, it's water-based in the cell. It looks a lot like a raw egg. It holds the organelles or parts in the cell to thrive and function within the cell. So cytoplasm is very, very important. And we learned about tonicity, what happens if the cells are in a different kind of solution. The nucleus is the brain of the cell, right? It dictates the activities of the organelles. It's kind of like the mastermind. It's double-walled. It has a nuclear membrane. It has chromatin, which is your genetic material that contains the DNA and becomes chromosomes. Nucleolus is a spherical body inside of the nucleus that contains the RNA that forms ribosomes. DNA and RNA. DNA is double strand, nucleic acid, has genetic code on it, chromatin. RNA is single-stranded, and it provides the ladder for protein synthesis. We won't go too far into that, but it forms proteins. Your ribosomes, organelles are found. So ribosomes are made of RNA, and they're a cell remodeler. They maintain and repair cell structure. So ribosomes are necessary. If they're found on the endoplasmic reticulum, which you might remember from biology, then the endoplasmic reticulum is called rough. Sometimes they're floating in the cytoplasm. Sometimes they're found on the ER, or the endoplasmic reticulum. Centrosomes are specialized regions in the cell that are needed for mitosis, or cellular division, or meiosis. Centrioles are involved in cell division. So basically they're like spindle fibers that will pull the cell into the shapes it needs to be for cellular division to happen. Centrioles are tubular, and they're found in pairs. Okay? They're located inside the centrosome. Your mitochondria, tiny bean-shaped. We think of that if we used the analogy of a school building. This is the furnace, the energy plant, okay? 95% of our body's energy, cellular respiration, which we just talked about, repair, movement happens here. These make ATP. ATP equals energy, and is adenosine phosphate. So the mitochondria, without mitochondria, we don't make energy. Here's your endoplasmic reticulum. Like I said, if it's found on, if ribosomes are found on there, they look like this with a bumpy surface, sandpaper. Protein synthesis happens here. A smooth ER has none, and that smooth ER, because it doesn't have ribosomes, is not going to be making proteins, it's going to be making lipids or fats. Your lysosomes are, I always think of Lysol. They're like kind of cleaning up, Lysol. They're breaking down, they're cleaning up, they're wiping away, they're getting rid of unwanted waste or bacteria. Phagocytosis is a process of a cell eating. So anything that's kind of waste product, it's cleaning up. Golgi apparatus looks like a flattened cell, like membrane sacs. It receives the proteins from the ER, that rough ER that's synthesizing proteins, and it's going to process it further. It's like a packaging plant. Okay. Salivary glands, pancreatic glands have a lot of Golgi apparatus because they have a lot of secretion or storage. So these are packaging plants. Okay. Your flagella, only some cells have these. There's like tails that whip around and help cells move. Sperm would be an example of this. They have a flagella. Cilia. Some cells have cilia or hair-like projections on their outer surface, and they move in wave-like motions. They might be lining like the nasal cavity, trying to get, smoke or dust or debris out of the nasal cavity. And here is a quick video.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, The respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Okay, so I thought that was helpful kind of to bring it all together. As you heard in the video, plant cells contain a cell wall, animal cells do not, so we do not contain cell walls. We have cell membranes, and that cell membrane has lipids with their heads and their tails, kind of tails facing in. It's a boundary. We call it a semi-permeable boundary because it allows some things in and other things it does not let in. So it's a protective covering and it's sort of a gatekeeper. And I'll play this video. I think this is the same video. Oops. Okay. Structures of the cell, again, we have the nucleus, the organelle, the cytoplasm, and the cell membrane. Okay, transport methods, active transport and passive transport. It's how things get in and out of that semi-permeable membrane. And if it's active, it requires energy. If it's passive, it does not. And remember, energy is ATP. So if you see something requires ATP, it is active. Again, the cell membrane is semi-permeable, so it allows some things in, some things not in. I kind of think of it as a bouncer. It's made of lipids and proteins, and it has, most of it is this phospholipid bilayer, meaning 2 layers. And it has, it prevents hydrophilic substances from going through, but hydrophobic can pass easily through. It identifies or what we call histocompatibility markers as it is coming from a certain person, and it has some channels that allow things into the cell. Okay, as you can see in this animation, some things like diffusion is a passive, requires no energy. Diffusion, just things float across that cell. If it's facilitated diffusion, like in the second picture, it has a protein in that phospholipid bilayer that kind of facilitates movement through. Still does not require ATP, so no energy. Diffusion is like making a powdered drink. Diffusion is the most common form, and a substance of higher concentration travels to an area of lower concentration to kind of even it out. Osmosis, whereas diffusion is talking about the substance, the solute, right? Going from areas of high concentration to low concentration. Osmosis is talking about the movement of water. So water travels through this selective permeable membrane, that's the cell membrane. Water moves from areas of low to high. It tries to dilute this too concentrated area, okay? So water will move from low to high until things are equal. That's osmosis. Okay. And I like this Amoeba Sisters animation because this shows if you have a cell, we talked about this earlier, if you have a cell in a hypotonic or low solute, low concentration solution, water is going to go and bloat the cell. Okay, it's going to go from areas of low concentration to high and bloat the cell. In an isotonic, it remains the same. In a hypertonic, if you've got too much concentration in your solution, the water from inside the cell is going to try to move out of the cell to dilute that solution, thereby shrinking your cell. So I like this animation because I feel like it visually shows you. All of this being passive, requiring no energy.
Before we take a look at how osmosis and tonicity affect a cell, let's review what each of these terms means. Osmosis represents the diffusion of water across a semi-permeable membrane. The term tonicity refers to the relative solute concentration of two environments separated by a semi-permeable membrane. In other words, by comparing the tonicity of the solutions, you can determine the direction in which osmosis will occur. To demonstrate how tonicity affects a cell, let's place some red blood cells into a beaker containing pure water. In this case, the solute concentration is greater inside the cells than in the surrounding water. In other words, the contents of the cells are hypertonic in relation to the hypotonic contents of the beaker. Because of this, osmotic pressure results in the diffusion of water across the membrane and into the cells. Over time, if enough water enters the cells, the cell membranes may burst. This is called lysis. Now let's place the red blood cells in a beaker containing a solution of salt, such as sodium chloride. Since the contents of the beaker are hypertonic in relation to the interior of the cells, the water within the cells will diffuse across the membrane and into the contents of the beaker. This causes crenation, or shrinking, of the cells. If the red blood cells are placed in a beaker whose contents match the tonicity within the cells, then there is no net gain or loss of water. The environments within the beaker and inside the cells are said to be isotonic or the same. An important thing to remember is that osmotic pressure always causes water to move from a hypotonic environment toward a hypertonic environment. In other words, water moves toward areas of high salt or sugar concentrations. This simple process is used to drive the operation of our kidneys and explain some of the physiological consequences of diseases such as diabetes.
Okay. So I like to think of, if we go back one, I like to think of the shrinkage of cells. Think about when you go to salt water, you go to the beach, your hands prune. That's because water is leaving your cells in your hands and going to the area of because it's such a highly concentrated salt solution. Okay, filtration is also a passive transport, but pressure is sometimes applied to force water or dissolved molecules across the membrane. So rather than like diffusion or facilitated diffusion, where it's just kind of floating, this has got some pressure applied to it in filtration. Again, your facilitated diffusion has a carrier protein, so it has an area of the cell membrane that this carrier protein allows that substance to diffuse more easily across. Glucose moves across the cell membrane this way. Okay, active transport pumps. Active requires ATP in the form of energy. So a lot of times a cell is trying to move a substance that's either too big or against the concentration. So it requires energy to move it across that carrier protein. Exocytosis is talking about when a large molecule is kind of, I think of engulfed or eaten. A large molecule, a toxin, cellular waste is moved outside and it's going to be kind of spit out into the cytoplasm. Your potassium and sodium ion pumps requires ATP. We're not going to get too much into that, but it does require ATP or energy to move against a concentration gradient. Endocytosis, endo meaning inside, is when it a certain cells can kind of engulf and suck in a substance, kind of make it into a vacuole or suck it in and bring it inside. That would be endocytosis, and that also requires energy. Here's another picture of like, so phagocytosis means eating or swallowing, and that's a form of endocytosis. So something, a solid particle comes across in this first picture, and the cell membrane kind of engulfs it and eats it. Pinocytosis is the same idea as phagocytosis, except that it's fluid. It's pino means drinking. So the cell will suck in fluid instead of a solid particle. And sometimes there's receptor mediated endocytosis. Don't worry so much about that, those specifics, but those are all examples of how something is moved across the membrane by endocytosis. Exocytosis, you've got a vesicle inside the cell and it kind of reaches the end of the cell and expels it. Active transport, again, requires ATP. So when you see ATP involved, you know it's active transport. Okay, the last section of this is talking about cellular division or cellular mitosis. And this is the process of making new cells. It's asexual reproduction, doesn't need someone else. We're dividing ourselves. And this is a function of eukaryotic cells, and our cells are eukaryotic. You have the cell cycle. And mitosis, or the cellular division part, is really just a small part of the whole cell cycle. Okay. Interphase is most of it. We have G1S and G2. That's interphase. That's all interphase. And the acronym for the cell cycle is IPMAT. So I for interphase, P for prophase, M for metaphase, A for anaphase, T for telophase. Okay, we'll go through this. Interphase is the majority, and 90% of that cell cycle is the cell lives in interphase. It's a normal functioning of the cell. It's preparing for division. It might be copying its DNA. It's making organelles. It's doing what it's supposed to do. It's functioning. And then that's the I in IPMAT. Now we have prophase, metaphase, anaphase, and telophase. This is the cell division part. And the first pictures are what it looks like under a microscope. Okay. So this animation I like. It kind of shows it. Okay. So let me go back to, it's going to cytokinesis is the last part where the two cells divide. So prophase, your cells, let me pause this. It's going a little fast. Cytokinesis, dividing. Prophase, okay. Prophase, pro means before. The nucleus disappears, the nuclear envelope disappears, and the chromosomes become visible. They anchor to guide wires of centromeres, the spindle forms. Okay, that's prophase. Meta, think of middle, okay, between. The chromosomes are going to start to line up in the center. That's what happens in metaphase. In anaphase, you have pulling apart. Those spindles are starting to pull apart. Telophase is the end. The chromosomes go to the far end of each two different, kind of look like two different cells. It's still one cell. After telophase, cytokinesis happens where the two cells cleave in half. And you have the division of the cytoplasm, and they actually kind of squeeze and pinch off. Oop. Let's go back and look at that animation now. So you've got metaphase, you've got, oh, it called it a, there you go, prophase. Okay, it's the nuclear envelopes are dividing. Cytokinesis, okay. Prophase, those, they're duplicating. Metaphase, they're lining up in the middle. They called it a chromosome parade. Anaphase, they move to the sides. Telophase, they're really moving to the sides and developing their own little areas. And cytokinesis is the cell is pinching off. Okay. Telomeres are non-coding strands at the end of the chromosomes. They protect the ends of the chromosomes. So sometimes mistakes in replication can happen with aging or mutations. Meiosis is different than mitosis. Meiosis is involved in sexual reproduction, whereas mitosis is asexual. So meiosis involves two different cells uniting to form a new cell. Meiosis produces gametes or sex cells, and we'll refer to this back when we talk about reproduction. Mitosis is I reproduce myself. I'm kind of cloning myself. It's asexual. It's an exact copy. Meiosis results in different cells. Okay. So in a basic, very basic overview of this. Okay. We'll have to send that out. Okay. All right. I'll be sure to look at those videos and make sure that you get those. But that is chapter 2. So I'm going to stop recording. I know that's a lot. You might want to go back and rewatch that. I'll make sure that you get the videos included in that PowerPoint that weren't working. And that's it.